Hi everyone, today I'll be showing you the effect of different sunscreens in protecting yeast cells from UVB radiation. So the purpose of this experiment is mainly just to see how well different types of sunscreens can protect yeast colonies from dying. Baker's yeast, which is known as Saccharomyces cerevisiae, is one of the most common model organisms in human or experimental studies. Since yeast is similar to cell division in humans, studies consist of cell division and cancer research. UV radiation is on the electromagnetic radiation spectrum between visible light and X-rays. The Earth's ozone layer protects us from most UV exposure except for UVA. As seen in the chart here, most of UVA goes to the ozone layer, while UVB only a little bit does, and UVC can't even pass it. UVB is partially absorbed by the ozone layer and can cause severe sunburn or cancer if exposed to it over long periods of time. Sunscreen is used for protection from the sun's UVA and UVB rays to prevent extreme sunburn and skin cancer. Sunscreen is labeled with SPF and a number that shows the strength depending on factors of a person's skin type and how long they'll be outside. And here they state some other factors. SPF stands for sun protection factor, and its numbers can go above 50, but today we'll only be using 30 and 50. The experimental plan consists of testing yeast cells under UVB radiation with the protection of SPF 50 and 30. There will be 3 plates that will have 4 sections, which one will be the control, which is the exposed part, and then there will be foil, and then SPF 50 and SPF 30. The plates will be placed under a UVB transilluminator for approximately 3 minutes to make sure it doesn't kill all the colonies because it is a UV sensitive yeast. This will determine which sunscreen can protect the most colonies from dying, as long as the cells are spread out evenly on each plate to provide the most accurate results. My hypothesis is, if yeast cells are protected with SPF 30 and 50 from UVB, then there will be more colonies with SPF 50 than with SPF 30. So when we're testing the yeast cells, first we have to start out with using a micropipette to get the yeast cells out of the tube and add at least 1 to 2 drops to each plate. Usually you put it directly over the plate and drop it right down. And then we're going to have to spread the cells evenly around each plate with a pre-sterilized spreader, as seen here. You can either use a disposable one or a glass one. And then we are going to place all 3 plates in an incubator at 30 degrees Celsius, where the cells can grow in about 1 to 2 days. And then finally, the plates will be placed under a UVB transilluminator for about 3 minutes. So here's the data for only the sunscreen for each plate. And as we see here, for the majority, yeast cells survive the most with the protection of SPF 50, except for in plate 2, which it could have been that the cells weren't spread evenly enough, so there might have been more cells where the SPF 30 was, and there might have been less cells where the SPF 50 was. And here it's all the data for each condition. As you can see, foil had the most colonies survive because that part, it was completely covered, and exposed had the least because it was the most vulnerable. So in conclusion, my hypothesis did end up being correct, which means that the higher the SPF number, the higher the survival rate of yeast colonies. The only exception for this hypothesis was for plate 2, but if you do the averages of the colonies, the average of the colonies survived, you can see that it was higher for SPF50 than SPF30. So for future experiments, we could use a different UV radiation like UVA or UVC, or you could use a different protection, like you could do lenses from sunglasses versus some kind of sunscreen. So yeah, that is the end of my presentation. Here's my references.